Hello everyone, so welcome back to this particular channel, and today we are back with the another session of genetics. Yes, so in this channel today we will be discussing about the gamete formation and the dihybrid cross, which we were not able to discuss yesterday in the yesterday's class, right? So I hope that you all are very eagerly waiting for the class, and let's quickly start the class. But before that, I will have an information for you if you don't know about the cell cycle and all those things. So we have published a one short cell cycle class in around 4 o'clock. So if you are having any confusion regarding the concepts of the cell cycle and everything, so what you need to do is that you, after this class, you need to go back and watch that videos and clear all your concepts regarding the cell cycle. Okay, now in the previous class of this genetics, we were discussing about the gamete formation. Yes or no, everyone? Yes, sir. We were discussing about the gamete formation. So let us start from there. We discussed about the monohybrid cross. In monohybrid cross, we got to know that 3:1 ratio is formed in the F2 generation, in which 3 plant is tall and 1 plant was short. And in F1 generation, all the plants were tall. And according to this, we discussed two laws which was given by the Mendel, which was what? The law of dominance and the law of segregation. All of these things we have discussed. Now let us quickly start with the gamete formation and complete the dihybrid cross. So how was the gamete formation, sir? The gamete formation we were discussing with the two methods. So basically, majority we are going to have one method for this gamete formation. What method, sir? The fork line method. Yes or no? Yes. The fork line method is the best method for the gamete formation. How, sir? How we are going to do? We are going to take a very simple example first. So let's have capital A and small a. Okay, capital A and small a. Now I would like you to identify that whether this capital A and small a, this is what, this is a gene. Okay, this gene is containing a similar kind of allele or it is similar kind of allele. So here when you are going to see that this gene is what, this gene is containing this similar kind of allele, that means one is what, one we have capital A which is dominant and the other one we have small a which is recessive. That means this is what, this is a heterozygous condition. Yes or no? Yes, sir. So whenever we have the heterozygous condition, what you have to do is that bifurcate it. What you have to do is that bifurcate it. Yes. Okay. Done. So here we have A and here we have small a. These are the gametes. Yes. That's all. So this is what we have got, gametes. Yes. Clear. Clear, sir? Okay. That's very good if it is clear. Then what, sir? Then the second case is that if suppose we have a similar kind of thing, okay, capital A and capital A. So if I am going to ask you about how many types of gametes here you are going to form, listen to my question very carefully. I asked you that how many types of gametes you are going to form. So sir, in this case, if we bifurcate also, both the gametes is going to have same kind of allele. So the type of gamete here we are what? Only one type of gamete is formed. But in the heterozygous condition, two types of gametes is formed. Okay. The basic idea is clear to everyone. Now, let us move on and let's make a very good table. Okay. So I'm going to give you some genotype. Let's write here genotype. And then we are going to discuss about the number of heterozygous pairs. So whenever you are asking about the gamete formation through fork line method, you need to figure it out like how much heterozygous pairs you have. So number of heterozygous pairs. Okay, sir, we will get it. Next, we are going to write what? We are going to write the types of gametes. Okay, we are going to write types of gametes. Clear? Great. Next what, sir? Next we are going to discuss the genotype. So I am gonna give you this genotype, that is capital A and capital A. So capital A, are you able to see any heterozygous pair here? No sir, we are not able to see any heterozygous pair. So let's write here as 0. Okay, great. Now, how many types of gametes will be formed? Sir, there will be only one kind of gamete, because if there is a cell, let's suppose this is a diploid body and this diploid body is making the gametes. So one allele will go in one gamete and one allele will go in one gamete. So that means here in this case, both the gametes is going to have capital A and capital A allele. That means both the gamete is of same type. That means the type of gamete is only 1. Clear? No doubt. Number of gametes will be 2. Okay. Number of gametes will be 2, but type of gametes will be 1. Clear? Okay, sir. Now, let's have some other examples. So, here I am going to give you few genotypes. Let's have a genotype, say, capital A, small a, capital B, small b. Okay? Okay, sir. Capital A, small a, capital B, small b. Now, what we have to do, sir? We have to find the number of heterozygous pair here. So, what is the number of heterozygous pair here, sir? See, we have capital A, where it is? We have capital A, small a. So, this is what? This is one heterozygous pair, yes. And then we have capital B, small b. So, this is one heterozygous pair. Clear? How many heterozygous pair in total, sir? We have what? We have 2 heterozygous pair in total. Yes or no? Yes. If now we have 2 heterozygous pair in total, then we are going to get how many types of gametes? We are going to get 4 types of gametes. How, sir? See here. Let's see. So, we have this capital A and small a, capital B and small b. Number of heterozygous pair is 2. Okay. So, one pair is what? One pair is capital A and small a. One pair is capital A and small a. Then again we have the another heterozygous pair is capital B and small b. So, again we are going to bifurcate here capital B and small b, and here capital B and small b. So, how many types of gametes we will get? See, this is going to make one gamete, right? This is gonna make one gamete. So, let's write here A, B. This is gonna make one gamete, capital A and small b. Yes or no? Now, this is gonna make another gamete, which is small a and capital B. And similarly, this is gonna make another gamete, which is small a and small b. So guys, how many types of gamete we got? We got total 4 types of gametes, and see, 4 types of gametes, that was our answer. So the fork line method is very, very easy to remember. Okay, sir. We want to do some more questions on the fork line method. Okay, I'll give you some another genotype. So let me think of another genotype. Let's have a little tricky thing. Okay, little tricky thing. Let's have. So the genotype which I'm gonna give you will be. Should I make it hard? Okay, a little hard. Okay, so we have capital A, small a, and then we will have small b, and then we will have capital C and small c. Okay, to avoid confusion, I am writing this small c as cursive. Okay, clear? Now, we are going to find out the number of gametes and types of gametes. Clear? Okay. How, sir? First of all, identify the heterozygous pair. First of all, identify the heterozygous pair. Sir, I can see this is the heterozygous pair. This has capital A and small a, different alleles. That means heterozygous. But sir, what about this? This is not a heterozygous pair. This is a homozygous condition. Yes. So till now we have only and only one heterozygous pair. Next we have what? This capital C and small c, right? Chotu c and bada c. So capital C and small c, again heterozygous pair. Yes or no? So how many heterozygous pairs do we have? Heterozygous pairs we have only and only 2. Okay, heterozygous pair is only 2. That's great, sir. That's very good. Now what? Now we are going to follow the fork line method. So see, here we have capital A, small a. We bifurcated it, right? We bifurcated it because in one gamete capital A is gonna go and in other gamete small a is going to go. But sir, what will going to happen with this small b and small b? Whatever happens, this each of the gametes, all the gametes is going to have small b and small b. There will be no capital B because it's not there. So instead of making the lines, I could write here as this simple, right? Instead of bifurcating it, there is no point of bifurcation. Why? Because B and B will only be there. There is no types of B. So we have just written like this. Clear to everyone? Then what, sir? Then we have capital C and small c. Then bifurcate. Capital C, small c. Capital C, small c. Which means what? Which means that in case of homozygous pairs, don't bifurcate it. Just write it as it is, right? Just write in front of this particular thing. Clear to everyone? Yes. Now, sir, how many types of gametes do we have? We have, see, A, B and C. So, capital A, small b and c. Then this will make capital A, small b and small c, right? Now let's see, you will see that each and every gametes will have small b only. Why? Because there is no capital B, right? So here we will be having small a, small b and capital C, and then we are going to have small a, small b and small c. So this is how you are going to make the gametes. Is it clear to everyone? I hope that this is pretty much clear and it's a very, very easy method. No confusions, nothing. Whenever in the exams you're having any type of confusion regarding the gamete formation, follow the fork line method. The best method you can follow. Okay? Yes, sir. Now let us move forward and let us go to the dihybrid cross. Okay, sir. Now, if I talk about the dihybrid cross, so can anyone define the dihybrid cross? Why we are doing the dihybrid cross? Is there any what is the purpose of dihybrid cross? So that purpose of dihybrid cross is what? Is to study the inheritance of pairs of characters. Sir, explain it in a easy manner. Okay, let's suppose that you are going to study dihybrid cross on me. Okay, so you saw that my hair color is black and my eye color is, let's suppose, black. Okay, so now you're going to see that whether this hair color and this black eye color will be a same for my son, or whether this hair color or black eye color is kind of related, or whether how this hair color and black color will go and express its phenotype in my son. So if you are taking two characters at the same time and you are studying the inheritance and the heredity of the two characters simultaneously, then we do the dihybrid cross. So what's the purpose of dihybrid cross, sir? The purpose of the dihybrid cross is what? To study the inheritance pattern of a pair of characters. Okay, study inheritance pattern of what? Inheritance pattern of two characters. Yes or no? Two characters. Now, since this is a Mendelian cross, so the cross on which the experiment which we will be doing will be the Pisum sativum. On the Pisum sativum, we are going to do this particular kind of experiment. Okay, sir. Now, what we are going to do, how we are going to do? See, so the Mendel took two plants and he observed that one plant was what? One type of character for the seed color was, he observed that there are varieties of plants and there are two varieties of color. Okay, sir. So seed color was what? In some plant, the seed color was yellow. in some plant the seed color was yellow. Okay, so and in some plant the seed color was green. Okay, so got it? Yellow and green. Which was dominant? Remembered fat, Ria, go to village and yawn, which we discussed in the first class. So yellow is what? Yellow is dominant and green was what? Green was recessive. So if it is dominant, how should I write? Should I write with capital Y? Yes sir, definitely we should write with the capital Y. So I am writing this yellow as the capital Y. I am representing yellow as the capital Y. Okay. Now what? Now we have green. So green color, can I write small g? No, we can't write small g. We have to write it as small y. Okay, to avoid confusions, I am going to use italics for this particular thing like this and cursive for the small letters. Okay, this clear? Okay, sir. Now, what is the other character? Since we are discussing that this is a dihybrid cross, and in dihybrid cross we have to take care of the two characters. We are studying the inheritance of two characters. So one character to sir, we got side color. Other character, Mendel chose what? Mendel chose the seed shape. Other character which was chosen by Mendel was seed shape. Okay sir. Now what sir? Now here we can see that the seed color, the seed shape comes in two categories, two types. What? One category, one type is the round, and the other one is what? The other one is the wrinkle. We already discussed 7 contrasting characters, right? So in those 7 contrasting characters only we are having these kind of characters, right? So round and wrinkled. Okay, so round and wrinkled we have. Now for the round thing, we are going to use capital R. For the round thing we are going to use capital R. And for the wrinkle, what should we use? For the wrinkle thing, what should we use? Say, for the wrinkle, what should we use? Should we use small r? Should we use the small w? We will going to use small r. Yes, because recessive is small r. Whatever the alphabet you are using to denominate the dominant character, the same alphabet, but in lower case you are going to use to represent the recessive characters. Okay, sir, that's very good. Now, let us move on and let us understand this in a bit more detail. So, to do a cross, what do we need? To do a cross, we need parents. Obviously, without parents, there will be no cross. So, what things you have to do? First of all, take out the male and female parents. Now, Mendel was the person who wanted to have the pure line. Now, sir, what is pure line? We discussed about the pure line, but still I am making you revise once again. So pure line was what? He wanted the homozygous parents. Okay, homozygous parent. So how did he create it? He created this homozygous line by self-pollination. He continuously did self-pollination. Let's suppose a plant is tall and continuously that plant is doing self-pollination. So there will be a time that small t, if that plant is having the small t, that small t will also be removed. Okay, so small by self-pollination, after multiple times they got the pure parents. Okay, so here they have got two parents. One parent is dominant for all the characters. One parent is dominant for all the characters like capital Y and capital R. And one parent is what? One parent is recessive for all the characters, which means small y and small r. Okay, so this capital Y and capital R, which is dominant for all the characters, is what? Is basically a plant which is having yellow seeds and yellow color seeds with round shape. Okay, and the ones which is of small y and small r is having what? Is having recessive character, which is green color and wrinkled seed. green color and wrinkled seed. Okay, so we got it. We understood about the yellow round and green wrinkle. Now what? Let's do the cross. And since you are now able to understand how to form the gametes, so let's form the gametes. Okay. So gametes we are going to make with the help of what? With the help of the fork line method. So capital Y and small r, capital R and small r. How many types of gametes will be formed? See, if I'm going to write here, both are what? Both are homozygous pairs. And both are homozygous pairs, then how we can write here? For the gametes, we can write here that this is will be also capital Y, this will be also capital Y. And here what we are getting? So basically the gametes which we are getting, what? The gametes which we are getting is nothing but capital Y and capital R. So here we have the gametes, capital Y and capital R. Next what, sir? Next this you are going to tell me in the comment section how this particular small y and small r is going to get the gamete. They are going to have this type of gamete, which is small y and small r. Okay, clear? That's great. Now what? Now they are going to fuse. The gametes are gonna fuse. See, the gametes are gonna fuse, and here what kind of progeny we are going to have? We are going to have this kind of progeny, which will be having capital Y, capital R and small r. Okay, clear? What we will be having? We will be having capital Y and small y and capital R and small r. So this progeny, since this is the first progeny, so what is this? This will be known as F1 generation. This will be known as what? F1 generation. Clear? Yes, sir. It is very much clear to us. This is known as F1 generation. Now, after this, what we saw, that all the progeny, whatever the progeny we got, all the progeny is what? It is dominant in its character. That means all the seeds, whatever we are getting in F1 generation, they are all yellow and round. They are what? They are all yellow and round, right? Yes, sir. They are all yellow and round. Clear? This much is clear. This was the F1 generation. Now what? Now Mendel did cross between the siblings of F1 generation. Cross between what? Cross between the siblings of F1 generation. That means self-pollination. Okay. So Mendel did self-pollination. What was that? We got capital Y small y and capital R small r. Okay. Now next is what? Next we have, since we are doing the selfing, so we will be having the same kind of gene for the different parents. Like we are doing the selfing between whom? Between the siblings. So siblings will have the same genotype. Yes, sir. Okay. And then we are going to see that how they are going to form the gametes and how much progeny they are going to have. Okay. Great. Now let's make the gametes. Let's make the gametes. How you are going to make the gametes? With the help of fork line method as discussed. So here we have capital Y, small y, capital R and small r, right? So homozygous pairs are there. Yes, homozygous pairs are there. No, homozygous pairs are not there. Heterozygous pairs are there. So how we are going to write? We are going to bifurcate the heterozygous pair, which is capital Y and small y. Done? Yes, sir. Done. Till this we have understood. Okay. Now what? Again, if you see this capital R and small r, this is also heterozygous. So again, we are going to bifurcate. So this will be what? Capital R and small r. Again, this will be what? Capital R and small r. Yes. So this is how we are going to make the gametes. And now let us arrange them and find out how many types of gametes we have got. So for this, just give me 1 second. I am not able to select this. Yeah. See, magic done. Now, how many types of gametes? This one, capital Y, capital R will make one gamete. Okay, sir. We have capital Y and capital R. What about the next? We have this one, capital Y and small r. Okay, sir. Capital Y and small r. What about the next gamete? We have small y and capital R. So should I write here small y and capital R? Yes. And this is, can you see this one? Small y and small r? Yes. So we have small y and small r. In total, how many types of gametes? We got 4 types of gametes. Similarly, for this also you are going to get 4 types of gametes. Yes or no? Clear. Now, what we have to do is that we have to do the cross between these things, right? So if I'm talking about in total how many gametes you will be having, 4 from this parent and 4 from this parent. So total 8 gametes. We have to make the cross. See, to make the cross, it will be very pathetic, it will be very hard, it will be very confusing if we do like this, like capital Y, capital R, and then again we will take that capital Y, capital, and manually if we are going to do without following any rules, we will make mistakes. That will create confusion. So that's the reason why Mendel has developed Punnett square, which we also did in monohybrid cross. So if you have not seen my monohybrid cross lecture, what you have to do is that you have to go back to the video after this class. Don't leave this class. After this class, you go back, search the monohybrid thing in the playlist, and there you watch that video. Okay. Now, on Sunday, I'm having a big quiz session at 11 o'clock so that we can have crystal clear concepts for the cell chapter. We have completed one short session on cell chapter. If you need the topmost questions from the cell chapter, do attend that class. Okay. Now let's have the Punnett square. How to make Punnett square, sir? We don't know. Don't worry, I am here. I'm going to help you. Total, how many gametes? One parent is making 4 types of gametes. Okay, sir. So let's write here. I'm going to decrease the pen thickness little bit so that all the content will be here in the single page. Okay. So we have capital Y, capital R, 1 gamete. Write it. Okay, sir. Next we have capital Y, small r, 1 gamete. So let's write that also. Capital Y, small r, written, sir. Next we have what? We have small y, capital R, this written, sir. Next what we have? We have small y and small r, written. Great. Okay, done. Now we have another parent also having the same gametes. Yes, sir. So let's write those here. Capital Y, small capital R, capital Y, small r, and small y, capital R, and small y, small r. Just what I did, I just arranged the gametes. What I did, I just arranged the gametes. Nothing else I did. Okay, very easy. Then what? Make a table. Let's make a table. Okay, so give me a second so that I can make a table. So here in this, you don't have to remember also that how many boxes we have to make, sir, what we have to do, nothing. Just arrange the gametes. That's all you have to do, right? Now see, let's make some more. Oh, what happened? No, don't cut this. This way we don't have to make the gametes, right? Okay, now I'm going to draw one line here, one line here. Clear? Then we have one line here, again one line here, and one line here. Okay, so a little untidy table we have made. Okay, no issues. We are not here to make a tidy table. We are here to understand the concept of a dihybrid cross. That's great. Now what, sir, what we have to do? It's now very easy. Half of the work is over. You just have to mix and match now. See, capital Y, capital R, capital Y, capital R. Just add it. Y with Y, R with R. Okay. So how to do? Capital Y, write this capital Y and this capital Y. And then write this capital R, this capital R. See, is it done? Yes, sir. We made the table. Okay. Similarly, this capital Y you write here Is this a capital Y? Yes, sir, this is also a capital Y. So let's write here capital Y. This is what? This is capital R, but sir, this is small r. So yes, so we are going to write capital R first. Remember, whenever making gametes or progeny, the dominant one will, if dominant is present, then write the dominant first. Okay, then the small r. Written, sir. Here capital Y, see, here capital Y, here small y. So I am going to write capital Y and small y. And then what we have, this is capital R and this is capital R. So yes, just write the capital R and capital R. Clear? Next we have capital Y, small y. Let's write. And then we have small capital R and small r. Done? Yes, sir. It's done. Next what? Next we have small y, capital Y. So small y is coming here, but don't worry, you have to remember one thing, always write the dominant one first. So let's write the dominant one here first. So I have written dominant one, this, which is this, and then I have written, oh, this was also a capital one, right? We have this capital, okay, sorry, this is the capital one also only. So this is what? Capital Y and capital R and small r. Clear? In a similar manner, we are going to fill this table. So I am going to fill it very quickly so that we can save a little time. Clear? Okay, sir. So capital Y, small r. Then what here? Capital Y, small y. See, capital Y, small y, capital R, small r. Done, sir. This is capital Y, this is small y, this is small r, and this is small r. Done. Great. Now next what? Let's have, this is small y and this is capital Y. So capital Y and small y. This is capital R and this is capital R. So we have written capital R, capital R. Now what? This is capital Y, this is small y. So capital Y and small y, and this will be capital R and small r. Written? Yes, sir, written. Now what, sir? We have small y, okay, and then we have small y. So small y, and we have capital R and capital R. Great, sir, written. Now small y, okay, capital R, small r, written. Okay, done. That's great. Now what, sir? Now let's fill up the last one also. So this is capital Y, this is small y, and this is capital Y. So capital Y and small y we are going to write. This is small r and this is capital R. So again capital R and small r, written. Great. This is what? Oh, yeah. This is what? Small y, and this is what? Capital Y. So capital Y and small y, and this is small r and small r. So we are going to write small r and small r. In this two things, what we are going to write here? Small y, small y. Okay, sir, small y, small y. This is what? This is capital R and this is small r. Written. Great. Very good. We have written. Again, we have capital small y. Written, and we have small r. Done. This is what? This is our Punnett square for the dihybrid cross in for F2 generation. How F2, sir? How F2? See, the first one we got this one, right? This is what? Let's suppose this is their grandparents. Okay, grandparents had their son had their sons. Okay, so grandparents, son. Okay, grandparents, your father, which is the F1. You are the F2, the second generation. Clear? So this is how we got them, F1 and F2. Now we have to find the ratios here. That is a kind of a tough thing for us. Yeah? No tough, because I am going to make it easy peasy for you. How, sir? See, we will make a imagination. Okay, just imagine a right angle triangle like this. Just imagine a right angle triangle like this. 1 second. Not like this. Like this. Okay, a right angle triangle. I'm not able to make a triangle. Oh my god, what is this behavior, sir? See, like this. Okay, so what we're going to do with this triangle? See, we are going to make this triangle here. wait, I'll take white color. Yeah. So can you see this line? Yes, sir. Can you see this line? Yes, sir, we can see. And can you see this line? Okay, one second. Yes, sir. Now what? What about this triangle, sir? This triangle represents all the progeny with dominant phenotype. All the progeny with dominant phenotype. And count the number. Let's count the number here. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 progenies are what? 9 progenies are dominant. So let's write here. 9 progenies are dominant. And if it is dominant, sir, what will be their characters? That means yellow, yellow and round. The character will be yellow and round. Clear, sir? Next what? Next we have, let's imagine a cloud. Let's imagine a cloud which is what? Which is present adjacent to the triangle, which is present adjacent to the triangles. Okay. See, this is what? This is a cloud which is just near the triangle. Okay. Let's make here. Now, this is what? This is a cloud which is just near the triangle. Okay, sir. And this is the cloud which is just inside the triangle. Okay, like surrounded by the triangles, just near or inside the triangle. Okay, sir. Now what, sir? This is what? These are the seeds which are dominant for color. These are the seeds which are dominant for color, but they are recessive for the shape. So I can write here 3 of them is what? 3 of them is yellow. 3 of them is yellow and wrinkled. 3 of them is yellow and wrinkled. Clear? Clear? Yes? Yes? Okay, that's great. Now, again, what? Again, just remember one thing. This one. This is the odd one out here. How, sir? This is the only person. This is the only person which is what? Which is having genotype, which is having a full recessive phenotype. Okay. So let's write here 1, which will be what? Which will be green and wrinkled, which will be green and wrinkled. And what about this thing, sir? This thing will be what? This will be green and round. This will be green and round. Clear? So this is how you can remember the Punnett square very easily. So whenever you need to make a Punnett square in exam, if you are going to do like this, capital Y, capital R will make this thing. No. That will say, that will like, you know, you will take 5 minutes in making the Punnett square only. If needed, just remember, the last box is having what? Small y, small r, small r. Write it. The triangle is having what? It is having the dominant ones. Okay, all the dominant ones. Inside a triangle, we have what? We have a dominant for one character, which is what? Which is the seed color. Okay, and rest we are having the dominance for the seed shape. Clear? This is how you can make it. Clear, sir? Now, if I talk about one more thing, like suppose that we had the parental types were what? The parental types were having the combination with yellow, round and green and wrinkled. That means what? That means dominant with dominant and recessive with recessive. See here. If we go back in little detail, parental type in dihybrid cross were having the combination as dominant with dominant, right? Dominant with dominant. Yes, sir. We saw that yellow was with round and recessive was with recessive. Okay, recessive was with recessive. Sir, that means, is it true that dominant allele, all the dominant allele will be in same gamete and all the recessive allele will be in same gamete? No, it's not true. Why, sir? How can you say this? See here, we have the combination, new combination, yellow and wrinkled. Wrinkle was recessive, yellow is dominant. That means yellow and dominant, dominant and recessive is also present in the F2 progeny. See, this is dominant, this green is recessive, and this is what? This is dominant. So again what? Again we can say that there is no such relation between the dominant and the recessive allele. They express themselves without the relation, without depending on the in each other. So this is known as what? This is known as law of independent assortment. Is it clear to everyone? This is known as what? Law of independent assortment. Clear to everyone? That's very good. Okay, if this is clear, let's do some questions. Let us do some of the questions. Okay, great. So now let me have a very beautiful questions for you. So let's suppose I'm going to give a very basic question. Okay, in human beings, okay, the question says that in human beings, let's suppose brown eye color is dominant. Okay, brown eye color is dominant. Brown eye color is what? It is dominant. Great, sir. So what? And over what? Over the blue eye color. Okay, or I should write like this. See here, I'm giving you B for brown, and B for blue. Okay, now you have to figure it out which one is dominant and which one is recessive. So obviously the one with capital letter is dominant and the one with small letter is recessive. Okay, sir. Now what? Now they are saying that brown eye parents have a blue eye child. The parents are what? Brown eye parents. Brown eye parents is having a blue eye child. Is having a blue eye child. Okay, sir. Great. Now what? Now my question is that what can be the genotype of the parents? What can be the genotype of parents? Yes. What can be the genotype for parents? Should I give you the option? Definitely, sir. NEET also gives the option, so you have to also give the options. Okay, I'll give. We have capital B and capital B small b. Okay, then we have capital B small b and we have capital B small b. Then we have capital B small b and small b, and then we have capital B and small b. Giving you a second to think about it and answer me. Okay, giving you a second to think about it and answer this particular question. I'm getting aside so that you can have a look over the question. The question is this one. We have four options: capital B, capital B, capital B small b. We have capital B small b, capital B small b, capital B small b, small b, and we have this one, small capital B and small b. Okay, pause the video. No, don't pause. I'm giving you the time. Think about it and let me show some answers in your comment section. Okay, let's get back. I hope so. You have some of the answers in your mind. Okay, let's solve this question. Let us solve this question. So here, sir, according to this question, we have something like the parents is producing a recessive character. Okay, the parents is producing the recessive character. So can this produce a recessive character? My question is this combination able to produce the recessive character? No. How? Wow, sir, how you decided? See, capital B, They are having what? They are having capital B. How they will have a combination of small b and small b? No, it's not possible. How, sir? Make the gamete. If you are in confusion, make the gamete. Na? This is what? This is the condition. Right? Make the gametes. In all the cases, it is going to give you capital B. Every case is capital B. Okay, sir. This is going to give you two capital B and small b. Okay, now you're very happy that sir small b to aa gaya. But if it's there is the small b, but it is not going to give you the small b kind combination. How, sir? See, this we are going to get small capital B. Okay, and this we are going to get capital B and small b. See, we have what? We have the heterozygous condition here, and capital B was dominant. No blue color, brown color only. Yes or no? Yes, sir, we got it. We understood it. So this one is wrong. This one is wrong. What about the second question, sir? Second option. Okay, let's us check that. Capital B, small b, and capital B, small b. Clear? Now here what's happening is that, here what is happening, we have two types of gametes. Each one of them is gonna produce two types of gametes. Capital B, small b. Capital B, small b. Clear? Okay, sir. Now what? Now there is a chance of having that small b combination. And this is going to give, oh, what I have done, just a second, what is this behavior, sir? Wait a second, yeah, capital B and capital B. Okay, now capital B and small b. Okay, I'm very happy, sir. Next, what? Next we have this small b and capital B. This small b and capital B. Okay, and sir, see, this is the point what we needed. Small b combination is made, sir. This will be the answer. That's correct. This will be the answer. So now what you have to do is that you have to check about these two things, whether this can be the answer or not. Can we have this as the answer? Think about it. No, sir, we can't. Why? Both are dominant. So wrong. What about this, sir? What about this? What about the combination C? So if I talk about the combination C, let's see whether this can be the option or not. Combination C. Okay, we have what? Capital B, small b, and one second. What happened? Yeah. Capital B, small b. What combinations do we have? We have capital B, small b, and what we have? Small b and small b. Okay, so we understood capital B, small b, and small b and small b. Let's make a gamete quickly. Capital B, small b, and at any case it is going to give you small b only. Okay, okay, great, sir. Now we are going to mix and match. This is going to give what? Capital B, small b, and this is going to get what? Small b and small b. Yes, so in this case also, this thing is possible. But can I choose this as an answer? Can I choose this as an answer? No. Why, sir? Why? You're doing a scam? No, I'm not going to do a scam. See, brown parents, what it said, the question said brown parents. That means this does not represent the brown parents, right? This means the parent is itself blue. So this can't be the answer. I hope that this question is very much clear to you all, and this ends our session. So in the next class, we will be going to cover again lots and lots of questions. First we are going to do, and then we are going to start with the post-Mendelian genetics. And don't forget to attend the class at 11 o'clock on Sunday, in which we are going to do lots and lots of questions on cell cycle. And if you have not learned the cell cycle, if you have not watched the lecture of cell cycle, go back to the homepage of this channel and watch the one-shot sessions. and then let's come and meet at 11 a.m. on Sunday to have a mega quiz session. Those questions will be those type of questions which is going to appear in NEET 2024. So till then, bye bye everyone, and see you in the next